All right. So it's the immune system's job to remove foreign proteins, and this matters a lot to us at Genentech because antibody drugs are foreign large molecules. And so if you have an immunogenic response against an antibody drug, you'll reduce the efficacy, or worse, you'll cause safety issues that aren't found until very late in a clinical trial. And so we'd like to avoid that. And so one of the main pathways that the immune system uses to identify these foreign proteins is through peptide presentation by MHC class 2. And so an antigen presenting cell will come over, it'll eat up protein. That protein will get cleaved into peptides. Some of those peptides will be bound to MHC class 2, where some will make their way up to the cell surface and be presented. And then a T cell can come in and recognize this complex, and if it deems it foreign, it'll launch an immunogenic response. And so the idea here is, if we understand peptide MHC class 2 presentation, we can reduce the presentable peptides that are in our antibody drugs, and we can reduce immunogenicity. So MHC class 2 has some tricky elements for modeling. It's a highly polymorphic protein, so thousands of these alleles have been identified. Each person can have up to 12 alleles, and our data set is highly multi-allelic. And then finally, there's this open binding groove that allows the peptide to translate within it. And so you both have to know what sort of patterns the allele is looking for, and then you have to be in a binding core, and then you have to be able to identify a binding core in a longer peptide. Fortunately, though, we have an amazing public data set with lots and lots of examples over a good spread of alleles. There's some considerations that you have to make in generating the data, but we make those considerations. And so for modeling, I think it's pretty intuitive that a graph neural network approach is going to be well suited for the problem. It's a three-dimensional binding problem, and there's a lot of sort of reused residue-residue interactions, and so the inductive bias of a graph neural network should do a good job. The challenge here, right, is in generating the adjacency matrix. You're obviously not going to get that from crystal structures unless you want to spend an eternity getting, a million of these. But fortunately, with AlphaFold 2 multimer and other techniques, you can now simulate structures between peptide and MHC class 2. And it does a reasonably good job, but it tends to overpredict adjacencies. And then for a subset of structures, it actually does a pretty poor job predicting. And this is on training data. And so instead of collecting a structure for every single peptide allele pair, we decided to just make like a canonical adjacency matrix for each gene, DR, DP, and DQ. And so you get a lot of structures over the alleles in a given gene, and you align the binding cores to get an aligned adjacency matrix like is shown here. And you see that there's a lot of common conserved adjacencies even over different alleles. And so just choosing the common adjacencies leads to this canonical adjacency matrix. And then you can just rely on the model to ignore for a specific peptide allele pair which adjacencies shouldn't be there, and to create some interactions between residues which are missed. So, we need to identify where the binding core is to create the right graph. And in doing this, all we do is just generate every possible graph for the sliding window of binding cores. We call that graph enumeration. And we'll just have the model choose the correct one during training. And so now we can sort of create the skeleton of the model. You have your sequences come in, they get embedded, positional encoded. You enumerate all these graphs, you do some sort of message passing, some sort of graph readout to get a good graph embedding. You take that embedding and you get a likelihood. And so you'll have that for all these different graphs. You'll choose which one was the best, and you'll calculate your loss with respect to that. So in practice, graph attention ends up being a good message passing, and then an attentive GRU readout for creating the graph embedding perform the best. And so now we have a really powerful sequence-based model at the end of our GNN, and so it's sort of a question of how much work is really being done by the adjacency matrix. So you can see that the 9-mer binding core adjacency matrix performs the best. If you have an empty adjacency matrix or a full adjacency matrix, performance drops off quite a bit. And then if you have a poorly chosen adjacency matrix, like the NetMHC 2-pan adjacency, you're unable to get all of the performance that you would from a better chosen one. And so the model does a lot better than the existing state-of-the-art, which is great. And so now we can apply it to antibody immunogenicity to create a risk assessment. So we have a data set of 100 antibodies with their immunogenicity rate, which is really large for this type of data, but not really large enough to do any sort of training. So we just created a simple heuristic and just sort of evaluated it. So you generate peptides. Every peptide that can be generated from an antibody, you get binding core predictions for all these peptides. If you align the antibody to its nearest germline neighbor, and if the mutation or the change is is outside the binding core, you ignore those. If the binding core is observed a lot in OAS, a big BCR database, you ignore those. If the peptide was unlikely to be presented as deemed by the model, you ignore those. And you're left with just a few unique binding cores. And so you repeat this process over 8 common DR alleles, as is commonly done, and you add all those up, and you'll produce the plot at the bottom right. And so you can see that good separation is obtained with this heuristic for non-immunogenic and immunogenic antibodies. It doesn't matter too much if you use Graph-pMHC for this approach or NetMHC 2-pan, but both work better than Sapiens, a large language model trained on BCRs. And so what I think this is showing us is that having these binding cores in antibodies is bad. If you reduce them, you can really significantly reduce your immunogenicity risk. And I think with the ascendance of generating antibodies in silico, we should be able to generate antibodies that really have very few binding cores. And so I think in the future, immunogenicity will be much less of an issue in our clinical trials. So with that, I'd like to thank you for listening and ask for any questions.
Thank you. It's time for a question. Yes.
Thanks. So can you use the same method to predict epitopes for infectious disease? So positive epitopes instead of negative epitopes?
Yeah, absolutely. We care a little less about that at Genentech, but it's definitely something you can do. Yeah, and you could just run a screen through every epitope that can be generated from a virus or whatever and get a good idea of how likely, an immunogenic response will occur.